Drosophila melanogaster is a powerful model to evaluate in vivo the potential toxic and endocrine effects of chemicals, such as endocrine disruptors, that represent a serious problem for human health and natural environments.
Addressing the effect of EDCs on the hormonal life traits of the fly, such as fecundity, fertility, developmental time, and lifespan, it's an easy, cheap, and fast way to investigate endocrine disruption in vivo.
The fertility, development, and lifespan in Drosophila can be affected by several factors that have to be carefully controlled to ensure the successful outcome of this protocol. Therefore, maximum attention in rearing and handling flies is required.
To assay the taste of the selected EDC, after anesthetization, transfer 15 young flies from the fly bed to 4 parallel vials, containing cornmeal medium mixed with a food coloring. The control vial contains the solvent alone, and the 3 other vials are supplemented with different doses of the selected EDCs. Keep the vials in a humidified incubator for 1 day with a natural 12-hour light. After anesthetizing the flies again, transfer the immobilized flies to a white paper and place it under a stereo microscope to observe. Compare the abdominal coloring of each treatment group with respect to the control group. To assay the reproductive performance of the flies, prepare 3 vials of flies for each EDC as parental vials, with 8 females and 4 males in 10 milliliters of cornmeal medium supplemented with EE2.